And I am also going to get our slide deck going for today's class. So today what we are talking about is we are going to be talking about, I'm sorry, we're going to go into a discussion of enzymes, but we're going to be kind of applying it to a discussion of sugars. Specifically, we are going to be talking about the process known as glycolysis. And the reason that we're going to be talking about glycolysis is because, well, it's a central metabolic pathway. Um, and it's a really good one because we talk about lots of different enzymes and all of that. Okay, sorry, just 1 second. Submit, publish. Okay, so slide deck is week 8, day 1. I'm going to go full screen on this one. Here we go. Okay, so glycolysis. This is our pathway. This is, if you've ever heard of glycolysis, fantastic. Um, it's more than likely that if you've heard about any sort of metabolic pathway, you've heard about glycolysis, and maybe you've heard about the Krebs cycle, also known as the TCA cycle or the citric acid cycle. Glycolysis is a really great model to kind of learn other pathways. And we're going to kind of take a couple of different angles when we look at glycolysis. So, first of all, we have this molecule right here. This molecule is none other than glucose. And really, we bring up glucose because we're looking at the purpose of glycolysis. Why are we using glycolysis? Why are we studying glycolysis? And the reason for it is because what we want to do is we want to take this molecule right here, which could somebody add to the chat? How many carbons does this molecule right here have? We're going to take this 6 carbon molecule and we are going to split it into, well, could anyone add to the chat how many carbons are in this molecule down at the bottom? We've got 1, 2, 3. Okay, perfect. 3. So we're going from 6 carbons down to 3 carbons. Well, it kind of makes sense that we're actually going to go to 2 3 carbon molecules. Okay, so glycolysis at its heart is taking this molecule right here and making 2 pyruvates. Now, I always like to emphasize and stress kind of the carbon accounting because I think that it's an important part of understanding the process as a whole. So we're taking 6 carbons of glucose and breaking it into 2 pyruvate molecules. So what that means is at some point we'll be looking at this pathway and we'll be looking at a step, and that step is actually happening 2 times over. Okay, so this is what at its heart, what we are doing in order to make this happen is we're going to invest some adenosine triphosphate, some PI or inorganic phosphate, ADP or adenosine diphosphate, and NAD plus. If you've ever heard of NAD plus or NADH, these are electron carriers. I'm going to say E carriers. Could anyone tell me what type of reaction will you use, or do you think, or no, let me rephrase that. What type of reaction do you know of where you talk about the transfer, something gaining or losing electrons? Redox, perfect. So oxidation reduction reactions or redox reactions. So lots of what you're doing in glycolysis is to not only get ATP, but also to get this molecule right here, NADH. So at the end of glycolysis, we will have produced 2 pyruvate molecules. Okay, so that accounts for all 6 of our carbons, but then we'll also recover ATP and NADH. Okay, so NADH is an electron carrier, and so, that's going to be very powerful. Could anyone tell me in the chat, or you can unmute yourself and say this, if you had something like pure potassium or pure sodium, a group 1 element, if you took one of those group 1 elements and threw it into a cup of water, what would happen to it? And before you answer that, maybe if you want to, you could say what would happen to it kind of electronically with respect to the electrons that it harbors. So if we took potassium and put it into a glass of water, or just left it outside. Let me ask you this. Would it gain or lose electrons? Okay, we got gain electrons. So if we've got, I always like to think about lithium. Today, I'm not taking any polls for attendance. I got a private message asking if we're going to. And I am recording this, so I'll be posting this on Canvas later. Okay, so if we talk about lithium, well, lithium as a group one element, what it likes to do is it becomes Li plus. And in order to do that, it has to lose an electron. It gets oxidized. Now, so, okay, cool. Something loses an electron, other things gain electrons. Great, great. Fantastic. Interesting to know. Now, if I were to get pure lithium or pure potassium, that's exactly what I'm looking for. It explodes. It's a very violent reaction. So I want you guys to think about oxidation of something or something getting oxidized or redox reactions as a whole. I want you to think about them as, well, they release lots of energy. So I'm going to say redox reactions release lots of energy. Now, it's entirely possible that you're like, yeah, I got that. I understand that. But that's something that when you're talking about a biological system, it's a little bit more difficult to visualize because, well, we're talking about, transfer of electrons and okay, well, here's this voltage potential. But you don't have that like exciting, oh, wow, it's on fire or anything like that. And hopefully you don't whenever you're talking about a biological system. Okay, so here is our pathway as a whole. I want you guys to know these steps. I want you to know the structures. So whenever I say steps, I'm referencing the enzyme names. I want you to know what these enzymes do. I want you to know the structures. And whenever I say structures, I want you to think about them as, well, reactants and products. So the first step, for example, hexokinase. Well, it's going to take glucose and it's going to add a phosphate to it. And it's going to make a molecule known as glucose 6-phosphate. So this is a whole pathway, but what we're going to do is we're going to kind of look at it a little bit more step by step. Okay, so here's all the learning objectives for this as a whole. And in order to get into glycolysis and kind of make a little bit more sense of all of these different steps and process this information a little bit more linearly, what I'd like to introduce you to is this first learning objective. I'd like you to become familiar with the 6 different classes of enzymes. Now, could anyone add to the chat, throw a number or a letter in there, if anyone is going to be preparing for the MCAT? Okay. Now, in your preparation for the MCAT, unfortunately, I really hope that that means you're like, unfortunately, it's nerve-wracking. I just spent 74 hours studying for or preparing to register for it, as opposed to, unfortunately, I have to become an evil doctor. Anyway, sorry, bad dumb joke. My wife told me the other day that my jokes don't make sense. And I was like, okay, well, anyway. So if you're preparing for the MCAT, when you're looking at this list of different classes of enzymes, there is a 7th class. So there are 7 classes of enzymes. I don't really bring that one up. I will mention it, but I'm really focused on the first 6. So your 7th class, which I'm going to add over here, is, I believe they are the translocases. And the reason that I kind of shy away from that is because this 7th class is relatively new. I believe that the 7th class was kind of like minted, I guess you could say, in like 2016 or 18. And that 7th class, well, the reason that it kind of was not a previous class is because the translocases, what they do is they'll take a molecule from one side of a membrane and transfer it or translocate it or relocate it to another side of a membrane. Now, in that, there's lots of different reactions that are taking place in order for that to happen. Some of those reactions being oxidation reduction, transferase, hydrolase, lyase, isomerase, and ligase reactions. So it's almost like, well, the seventh one is really just it's built up by these other ones. Now, I'm not saying I don't believe in it or anything like that. I'm just saying I want to keep it a little bit more succinct. And so that's why we're talking about these six. So the six that I want you guys to know for this class are number one, your oxidoreductases. Number two, your transferases. Three, hydrolases. Four, lyases. Five, isomerases. And six, ligases. If you're preparing for the MCAT, you will have to know about the seventh one, the translocases. And like I said, they came around and or they were, I guess, agreed upon as a class in like 2016, 2018. Now, the ones that are circled are the ones that I want you guys to know about. We're going to get into each one of those a little bit. Now, before I do that, what I want you to recognize is this numbering system. We've got number one, and then there's 1-1 or 1.1, and then there's 1.1.1. What this all relates to is every single enzyme has this series of numbers. Could anyone add, let's say, a slash to the chat if you're familiar with an IP address? Something that your computer has. Exactly. Great, great. And if you're familiar with an IP address, you'll know that it is a series of four different numbers. It's something like this, and I'm just putting X in as a placeholder. So it's basically this. It could be like 192. Most folks have 192.168 because they're behind a router, .0.1. And that's the extent of my IT knowledge. But what I like to look at these numbers right here as similar to IP addresses because they give you some information. For example, this first grouping, this first number, that would be basically your big class. What type of enzyme is this? Oh, it's an oxidoreductase. Okay, so type. The second number, for example, would tell you a little bit about maybe the substrate that is used. I'll say substrate used. The next number would be maybe something about the product generated. And then the last one would be the specific. Joel, you didn't leave enough space for that. So I'm going to go all the way down here. Specific reaction. Okay. Now, I do not want you to commit these numbers to memory or anything like that. But if we look at what I'm kind of alluding to is see this number one, or actually I'm going to go with the number one, that's your oxidoreductases, and then this 1.1.1, that is an enzyme that acts on a CHOH group of a donor, and it uses NAD or NADP as an acceptor. And then there would be something like zero point 0.1, and that would be a specific enzyme catalyzed reaction. Could you give me a Y or an N in the chat if you follow all that? I always just kind of think about this group of four numbers as like an identifier for an enzyme. Now, like I said, I don't want you to commit these to memory. The folks who have good knowledge of these numbers would be the folks who study specific enzymes. Hey, I study this enzyme that acts on a CHCH group with NADP and it generates this product. That's where you're going to be very, very specific. So my enzyme is 1.3.1.4 or something like that. Okay. Now, here are our six major classes. And unfortunately, I need to move these reversible arrows up. Okay, so these reactions are reversible. The first number, the first class, is an oxidoreductase. So you're going to have a reactant like NAD, and you're going to have a product like NADH. This is reversible though. So a good idea is anytime you see a reaction that has NAD or NADH, you're looking at an oxidoreductase more than likely. Now, a transferase is a type of enzyme that transfers a carbon, nitrogen, or a phosphorus containing group. In which case you might be thinking, okay, well, what does that look like? Well, one thing that it looks like would be something like using ATP, adenosine triphosphate. Okay, so a very cartoonish model for ATP looks a little bit like this. A, and then P, P, P. The P with the circle is a phosphate group. So an enzyme might pick up this molecule and then basically pluck that phosphate off and transfer it to, I'm going to call it molecule X. This enzyme is going to transfer this group onto molecule X, and so the resulting molecule looks a little bit like this. So a transferase moves a group or moves something from one substrate to another substrate. And this enzyme, phosphofructokinase, is one enzyme that we will see in glycolysis. Now, lactate dehydrogenase is not an enzyme that we'll see in glycolysis, but it is related to um, metabolism and metabolism of sugars, in particular because what's one of our products? Pyruvate. Okay. Pyruvate and lactate. What's another term? Okay, so lactate is the ionic form of this compound. What's another term you can think of that might be important or relevant for lactate? What's another? I'm trying to think of what's the best way I can word this. When you're exercising and suddenly your legs get really tired, and it's not because of muscle strain or anything. What's the buildup? What's the compound that builds up? Lactic acid. So lactic acid is produced by lactate dehydrogenase. This is a reversible reaction. Okay, so that is one of the fates of pyruvate. Okay, so it's kind of adjacent to glycolysis. Our last enzyme is represented by the number of three, the lead number three, and that is urease. This cleaves hydrolases. A hydrolase is an enzyme that uses water to break a bond. So one corresponds to an oxidoreductase, two corresponds to transferase, three a hydrolase. Now, we will get into phosphofructokinase specifically after a little while. Our next group is led by the number four, a lyase. Cleaves a carbon-carbon bond and carbon-sulfur and certain carbon-nitrogen bonds. Isomerases, well, just takes isomers and kind of rearranges them. These two enzymes, aldolase and phosphoglucose isomerase, phosphoglycerate mutase, we will talk about these in glycolysis. And last but not least is a ligase. A ligase forms a bond between carbon and oxygen, sulfur, or nitrogen. The way that it does it is it breaks a high energy phosphate bond. So if you look at this, pyruvate, ATP, and CO2 are the reactants. The products are oxaloacetate, ADP, and PI. I leave that out there kind of I leave PI off just because I need it to be my like trigger reminder sort of thing. This molecule oxaloacetate is essentially pyruvate plus CO2. Now, in order for that CO2 to be added to pyruvate, well, you have to break up. You have to hydrolyze ATP in order to drive that reaction. So the enzymes or the classes of enzymes that we will encounter quite a bit in glycolysis are we'll talk about an oxidoreductase, transferase, a lyase, and some isomerases. So what I want you guys to remember for this is I want you to look at these numbers. The 1 for oxidoreductase, the 2 for transferase, 3 hydrolase, 4 for our lyases, 5 for our isomerases, and 6 for our ligases. And rather than trying to remember this definition, instead, when we look at glycolysis, try to think about, oh, well, that looks like this type of enzyme. I think that's a much more effective use of your time. And what I'm going to do is as we go through this list, I'll toggle back and forth so that we can kind of take a look at everything. Okay, so my takeaway from this is think about those names and numbers, but let's kind of project forward to applying those numbers to the different steps in glycolysis. All right, so glycolysis, it's kind of like your amino acids. It's a bunch of steps And one thing that people, or sorry, that's not like your amino acids, but it's a bunch of information that historically, like if you have any friends who have taken a biochemistry class or prepared for the MCAT, chances are they've said really supportive things like, oh my gosh, amino acid structures are going to eat your lunch. Oh man, you're in for it. Or, oh my gosh, glycolysis, oh, get ready. It's hard. Oh my gosh, it's so challenging. Um, if you haven't had any friends who said that, that's good. That means you have true friends. Um, this sort of has that reputation of being like, well, it's a lot of information and you just gotta work through it deliberately. And I don't know that I necessarily disagree with that, but I think that whenever I think of glycolysis, I think that there is definitely a work smarter, not work harder approach that you can take. Now, the first thing that you want to do whenever it comes to working smarter to understand glycolysis is to break it into two different phases. Just like we took our 20 proteinogenic amino acids and said, okay, well, there's some that are polar, some that are non-polar. The polar ones we can divide up this way. Oh, now I only have to remember two of them in this specific category. Then I can work backwards. Well, the two phases of glycolysis. Our first phase is commonly known as the investment phase. The reason that this first phase is known as our investment phase is because we use ATP. We invest ATP in this phase. And specifically, we're going to invest 2 ATP. Now, let's take a moment and look at these two steps. These two steps are phosphofructokinase and hexokinase. Okay. Now, I want to introduce you to the reactants and the products. Our first step, hexokinase, we're going to focus in on that. Our first step, the substrate is a molecule known as glucose. Our second step is a molecule known as glucose 6-phosphate. Okay. So thinking about that, the difference between glucose and glucose 6-phosphate, well, give me a Y or an N if you think it's reasonable to say that the difference is a phosphate molecule. Perfect. It is. Okay. Now, a fair question then is, well, how did it get there? Did it spontaneously appear? Or was there a molecule that maybe had that phosphate? Okay. Now, if we go back, we can see right here that ATP is one of our reactants. So when it comes to hexokinase, we really have two reactants. We have ATP and glucose. Now I want to take you back here and just look at these numbers. Oxidoreductase for 1, 2 transferase, 3 hydrolase, 4 lyase, 5 isomerases, and 6 ligases. Okay. Now, if we've got ATP as one reactant, and then we have ADP as one product, then our other reactant is glucose, and our other product is glucose plus a phosphate. How do you think, numerically, how do you think you would classify this enzyme based on those previous numbers? You got to vote for 6. You got another vote for 6. Now, if we take a step back, well, a ligase does require ATP. So too does a transferase. The difference between them is, let's look at our reaction here. Fructose 6-phosphate plus ATP, and then fructose 1,6-bisphosphate plus ADP. Whereas our ligase, this ATP is basically just broken up into two different things, ADP and inorganic phosphate. So what I'm getting at here is, yeah, we take this phosphate, this inorganic phosphate off, but then what do we do with it? We put it onto glucose to make glucose 6-phosphate. Does that make sense? Does that explanation make sense? Give me a Y or an N to the chat. So we're taking, yeah, sure. So we're taking ATP Let me show, I'll give you my quick cartoon model. So there's my A, P, P, P. There's my ATP right here. And then we're taking this phosphate right here that I put a circle around. And I'm going to change color there actually. I'm putting a green circle around it. We're taking this phosphate and we're going to put it onto glucose right here. And that's how we're going to get glucose 6-phosphate. So then the rest of this molecule will continue to exist as ADP. So with all of that said, what we're doing is, yeah, we're breaking a phosphate bond. It's a high energy bond, but we are transferring. That's a difficult color to see. I'm going to go with this one. We are transferring this phosphate from ATP onto glucose to make glucose 6-phosphate. So we would classify hexokinase as a transferase. It's a subtle difference between a transferase and a ligase. More times than not in glycolysis, we're going to see transferases and oxidoreductases. Those are our big ones. Can everyone give me a 2 in the chat if you can see how this is a transferase? Wonderful. Okay. Now, our next enzyme, phosphoglucose isomerase, also known as PGI. Oh, this one, before we look at the name, let's look at the reaction. We've got glucose 6-phosphate, fructose 6-phosphate. If you remember when we talked about glucose versus fructose, glucose, could you give me an A in the chat if this is an aldose or K in the chat if it's a ketose? What is glucose? Yeah, it's an aldose. What about fructose? Is that an A or a K? Absolutely right. Fructose is a ketose. I referred to fructose as the ketose version of glucose. So they're very similar. The difference between these two is basically that glucose is a aldohexose and fructose is a ketohexose. So they are both hexoses, both have 6 carbons. Now, with that in mind, and ignoring the name here, do these two molecules right here and here, do they have the exact same number of hydrogens, oxygens, carbons, and phosphorus? Do they have the same number of each atom? And I know that you're probably counting them all out right now, and you're like, just got it. Yeah, they do. So these two molecules are identical. But what's a difference about them? Or what is one example of a difference between them? Well, how many atoms make up the ring for glucose? 6 atoms. How many atoms make up the ring for fructose? 5 members. Okay. So if they're the same number of and identity of atoms, but they just have a different structure, well, we've just rearranged them. These two molecules are isomers of one another. And since they're both isomers of one another, our type of enzyme would be a isomerase. So our number would be 5, an isomerase. Can everyone confirm with an I that they follow that and they see that? Great. So one way that you can think about the steps in glycolysis is that we have a transferase, then we're going to an isomerase, or you could go with 2, 5. Okay, then what's our next step? Our next step is an enzyme known as phosphofructokinase. So what we are doing is we're taking ATP and fructose 6-phosphate. And then let's look at our next product, or our product of this reaction. FBP, also known as fructose 1,6 bisphosphate. So fructose 6-phosphate, fructose 1,6 bisphosphate. Could anyone tell me what the difference between these two molecules is? Well, if we look at carbon number one right here, carbon number right favor, you're absolutely right. There's two phosphates. Fructose 1,6 bisphosphate has a phosphate on carbon number one. If we look at carbon number one of fructose 6 phosphate, we don't have that phosphate on carbon number one. So basically, what that means is if you wanted to make fructose 1,6 bisphosphate from fructose 6 phosphate, all that you need to do is add phosphate. Can you give me a Y or an N to the chat if you follow that and you agree with that? Perfect. Okay. So we're adding it. Well, where's it coming from? Is it coming out of nowhere? No, it's coming from ATP. So essentially, we're transferring a phosphate from ATP onto fructose 6 phosphate. And in the process, we're making fructose 1,6 bisphosphate. So we've got, in terms of the sequence of our reactions, we have a transferase, then we have an isomerase, then we have a transferase. So 2, 5, 2. Is everyone good with that? Give me a 3 in the chat if you follow that so far. Okay. Now, our enzymes that do this are hexokinase. What does the isomerase do again? Great question. The isomerase is taking glucose 6-phosphate, this molecule right here, and making it, rearranging into fructose 6-phosphate. So it's taking a 6-membered ring to make a 5-membered ring. Not losing anything in the process, just making the ring a little bit smaller. Now, in terms of, well, why? Well, one thing that's doing is, let's look at carbon number 1 on glucose 6-phosphate. There's going to be some steric hindrance there, and so adding a phosphate to that is going to be kind of hard. But then when we rearrange the ring and make fructose 6-phosphate, now we've got this OH group right here that is super accessible and easy to add a phosphate group onto. Does that make sense? Give me a Y or an N to the chat. Okay. So after we've done these first 3 steps, we have invested 2 ATP. We've spent 2 ATP. What have we gotten out of this? Well, we've gotten 2 ADPs out of it, 2 adenosine diphosphates. The next thing that we want to do to conclude our investment phase is we want to break this molecule in 2. Break this 6-carbon molecule in 2. And so I always like to visualize it as I'm splitting it right down the middle. So carbons 1, 2, and 3, carbons 4, 5, and 6. That gives us 2 3-carbon molecules. And what is the final product that we're looking to make in glycolysis? Just a little bit of a hint, there's 2 of them. Pyruvate. How many carbons is pyruvate? 3. So are we done? No, but we're getting closer. Does that make sense? Y or N to the chat, please. So we're going in this direction where we're going to end up with a pair of 3-carbon molecules. Now, the 2 products that come out of this are, splitting this, are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These 2 molecules are both phosphorylated sugars. So that means that they are 3-carbon sugars and they each have phosphate. If you remember our names for our 3-carbon sugars, there was glyceraldehyde and there was dihydroxyacetone. So we've got glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Those are the 2 3-carbon molecules. That reaction that is done to take our 6-carbon molecule down to these 2 3-carbon molecules, that enzyme is known as aldolase. Now, could anyone tell me, not this bond, but what type of bond, like what atoms are connected by the bond that I've just circled right there? What 2 atoms am I really kind of focusing in on? I'm going to draw Well, not O and H. C, yep, a carbon-carbon bond. Okay, because I'm sorry, I was looking at this bond right there. Sorry. So I'm cutting right there, cutting a carbon-carbon bond. Ooh, and that's a covalent bond. And that's going to be kind of strong. So, what type of enzyme? I mean, the name of the enzyme that does this is aldolase. Let's see. Looking at our different options. So, is it an oxidoreductase? No, no indication there. Favor, you're absolutely right. A lyase, because lyases cleave carbon-carbon, carbon-sulfur, and carbon-nitrogen bonds. We're looking and needing this for the carbon-carbon bond. So now our sequence of types of enzymes is we have a transferase, an isomerase, a transferase, followed by a lyase. Okay. Now, where I just brought us to, or where we are right now, is glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Okay. So that's where we arrived at. But what I want you to notice is this step right here. So I'm put a star and now I'm drawing a box around it. Basically, the last thing that we need to do to conclude our investment phase is we need to take the dihydroxyacetone phosphate that we produced and make glyceraldehyde 3 phosphate. So both of these molecules are 3 carbon molecules. They each have a phosphate. So we're going to isomerize them. We're going to isomerize the DHAP, the dihydroxyacetone phosphate, this molecule right here. We're going to isomerize it to make 2 glyceraldehyde 3 phosphates. So my what I always like to highlight and stress is not stress. I hate saying that. But what I want you to walk away from the investment phase with is that we have a couple of different enzymes employed, a couple of different types of enzymes employed. We've invested 2 ATP, and maybe most importantly is we will end the investment phase, the first phase of glycolysis with glyceraldehyde 3 phosphate. The reason that I want you to walk away thinking, okay, I'm ending investment at GAP, 2 molecules of GAP, is because then you can look at the second phase of glycolysis and say, okay, well, I'm starting with GAP and I'm ending with pyruvate. I've got these 2 3 carbon molecules, and I just need to do a little bit of chemistry here, 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 and then I'm going to end up with a pair of pyruvate molecules. Or I could say, I'm going to take GAP and convert it to pyruvate, but I'm doing it 2 times. Could everyone give me a Y or N if that makes sense? Or, give me an N if you want me to repeat that. Okay. So if you're a fan of mnemonics like I am, I always think of the investment phase as transferase isomerase, transferase lyase isomerase. And that is a 5. Or 25245. I'm just trying to give some more information or more ideas of like how you can identify and remember this information. Now, another thing is I want you to know the sequence of the enzymes. And just so you guys know, this over here, I'm not going to have a question that would be something like, tell me the sequence of the numbers of classes of enzymes. I won't have that question, but I always think that it's helpful to, when you're trying to figure out how we go from one reactant to a product, I think that it's helpful to say, okay, well, I know my reactants, I know my products. Well, in the case of glucose 6-phosphate and fructose 6-phosphate, did I lose any atoms? Did anything get added? No. No bonds getting broken. I'm not taking one molecule and ending up with 2. Oh, that's just an isomerization. Okay, then, oh, now I got to narrow down what are my names of my isomerases. Okay, there's phosphoglucose isomerase. Okay, there we go. Um, I do want you to know the steps. Hexokinase and HK. What I always started out with was I remembered the acronyms. So I'd go HK, PGI, PFK, aldolase, there's not really one, and then TIM, or triose phosphate isomerase. So those are my steps in the enzymes that catalyze those steps. All right. Now, poll question that's not being asked, but what is the best term used to describe the sugar found below? And to the right. Okay, we've got an aldose, ketose, trodose, or a pseudose. Well, glucose is an A, aldose. You're absolutely right. Okay, now we're going to go on to our second phase. And here's where it gets particularly hairy. But if you look, this slide deck has 35 slides. I wanted to introduce the investment phase on one concentrated slide so we could kind of keep it all together and so we could think about it as one glucose molecule produces two glyceraldehyde 3 phosphates. I've got slides for each one of these different reactions, both in the investment and the what's known as the return phase. On this slide, what I want you to really focus in on is I want you to know that this is the second phase. And this is commonly known as the return phase. Return, or it's sometimes referred to as the payback. And not payback in terms of like revenge, but instead in terms of, well, what are the products that you're getting? Okay. Remember, we started with two molecules of glyceraldehyde 3 phosphate. So that's where we start. Throughout this process, we're going to invest two NAD plus. We're going to invest a pair of inorganic phosphates or PIs. We're going to return. Okay. I'm going to start focusing in on our return because that's what we really want to know about. We want to return to, or we will return to NADH. We'll invest to ADP. We will return to ATP. We will return to ATP. And we're going to end with two pyruvates. Okay. Now, when it comes to glycolysis, it's pretty common for people to say, okay, well, what is your return and what is the net return? First of all, this molecule shown on the far left is glucose. We're going to invest one glucose. We're going to also, so you can kind of think about these as our reactants. We're going to invest a pair of inorganic phosphates, a pair of nicotinamide adenine dinucleotide or NAD+, and we're going to invest a pair of ADP. The key here is the net investment. Then what are we going to get out of this? Well, we're going to get 2 ATP, 2 pyruvate, 2 NADH, 2 waters, and 2 protons. Now, it's important to be able to reconcile this with what we've previously seen. And this word net, think about this almost like an accounting term. Like, I always think about glycolysis as, well, you invested some capital, you invested ATP. Could anyone tell me in the chat how many ATP we invested in glycolysis? 2. Perfect. Now, if you look at this slide right here, specifically right here, how many total ATP are we getting back? Add that to the chat. Total ATP. 4. Okay. So if we invested 2 and we get a total of 4 back, what's our net ATP recovered? 2. Perfect. So that's where that number is coming from. And so you can look at this and say, here's our overall reaction. Glucose right here, ADP, inorganic phosphate, NAD+. 2 ADP, 2 inorganic phosphate, 1 glucose, and 2 NAD+. Our output, and arguably what's most interesting about glycolysis are these 3 things right here. The net and interesting takeaway is that we got 2 NADH, which this is a high energy compound that, it's ready to be oxidized. And whenever it gets oxidized, it releases a ton of energy. Then we got 2 pyruvates. Then we got 2 ATP. And that is our net. Now, with that said, does anyone see any CO2 here? No. I mean, at best, you could look at the pyruvate molecule and be like, ooh, well, part of that looks like a CO2, but that's a carboxylate ion right now. When you completely break down glucose, your product is CO2. Now, if we look at this and just kind of a quick glance at these steps and these steps, give me a one in the chat if you saw any investment of O2, and give me a two in the chat if you don't see any. Yeah, there's no investment of O2 there. That's because all of this is done without the assistance of oxygen. So this would be considered your anaerobic respiration. Your aerobic respiration is the type of breakdown of nutrients with the help of O2. So that's where you're going to get a huge amount of CO2. Now, here is the summary of it all. These are your 10 steps in glycolysis, or the 10 enzymes in glycolysis. After this, we're going to go step by step. So the first step is a hexokinase, or it is hexokinase. You're investing ATP. You're getting a transferase reaction to produce glucose 6-phosphate. So we've added a phosphate onto carbon number 6, which is this carbon right here. Hexokinase does this. Magnesium 2 plus, a divalent cation, is important for this. We will talk about ions that are important for reactions and driving chemical reactions later on. But for the time being, you just need to know that magnesium is involved. Then we've got glucose 6-phosphate that undergoes a reaction, an isomerization, to produce fructose 6-phosphate. Did the phosphate change where it was? Nope. Still on carbon number 6. Still on carbon number 6. What happened was we opened up our ring, and now carbon number 2 right here connects to that oxygen, which is what we've got right here. So we just made our ring a little bit smaller, which made this hydroxide group more accessible. This was an isomerization event, or an isomerase catalyzed this reaction. And when that OH group is available, now all of a sudden, could anyone add or tell me what type of enzyme can act on this? On that fructose 6-phosphate? The type of enzyme that sees that and is going to act on it is transferase. It's going to add a phosphate. That's going to give us fructose 1,6-bisphosphate, also known as F6, or sorry, yeah, we isomerized it, and now we've got FBP, or fructose 1,6-bisphosphate. The enzyme that catalyzes this is a transferase. This transferase is known as phosphofructokinase, specifically phosphofructokinase 1. Now, I'm going to go back a second. Look at this step. Does that arrow mean anything? Does the singular direction of that arrow matter? Does it matter? Y or N? When we have an arrow like this, that means it's singular direction. Now, enzymes can catalyze reverse reactions, but you got to think about, we look at, let's see, where is it? This is a good example. Which way is favored? A, the production of G6P, or B, the production of glucose? Which reaction do you think is favored? Absolutely. And this reaction, really, we don't want that arrow there, just because this is favoring the forward reaction, favoring the production of glucose 6-phosphate so much that we kind of don't even care about the reverse reaction. So with that in mind, it's pretty common to look at step number 1, step number 3 as irreversible steps. 1 and 3, IRR, irreversible. Now, after that, after our second transferase reaction done by phosphofructokinase, we are going to split fructose 1,6-bisphosphate. I took it from the cyclical or the ring form of fructose 1,6-bisphosphate right here, and I linearized it. in this model. Aldolase is going to break this bond between carbons 3 and 4, which is going to give us glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So we get two 3-carbon molecules that are phosphorylated. Now, from there, what we have is our triose phosphate isomerase that's going to take our dihydroxyacetone phosphate, our ketose, and produce our aldose, glyceraldehyde 3-phosphate. Now, what's important for you guys to know about is that when we proceed into that second phase of glycolysis, we're using 2 molecules of glyceraldehyde 3-phosphate, or GAP. These 2 molecules of this aldose is going to be what's going forward. That's what we want to focus on. So we've converted a ketose to an aldose. Now, what that means is that this molecule right here is how we proceed, is what we proceed with. Glyceraldehyde 3-phosphate. The first step that we're going to do in the return phase of glycolysis is we're going to take our glyceraldehyde 3-phosphate, which is this molecule, and we're going to phosphorylate it. We're going to phosphorylate it. So we are going to use a inorganic phosphate molecule, and we're going to add that inorganic phosphate molecule to make 1,3-bisphosphoglycolate. Okay, could anyone tell me, how would you classify this enzyme? And before you answer, I want to take you back to, well, that would be super far, to this slide right here. What is the name of our oxidoreductase? Lactate dehydrogenase. When you see this name, dehydrogenase, I want you to think, aha, that's an oxidoreductase, or whatever enzyme is a oxidoreductase. What else are we looking for? Well, it's also got NAD plus and NADH. Could you give me a 1 in the chat if you follow that so far? Okay, now we're going to go back to that step that we're on. Okay. Our previous steps where we were adding a phosphate, we were talking about transferases. The difference is that this inorganic phosphate, PI, it's just floating around. It's just in the cell. So this is not coming from ATP. Give me a Y in the chat if you follow that. So it's not coming from ATP. It's just kind of free form. It's floating around. So we are not transferring this phosphate from ATP. ATP is not donating this phosphate. Does everyone follow that? Y or N to the chat. Okay. And because we're not taking this phosphate from ATP and putting it onto this glyceraldehyde 3-phosphate, this is an example of an oxidoreductase. So one of the points that I'd like to make is that to be a transferase, you need a substrate to donate a group like P in a circle, the phosphate in a circle. Does that make sense? Y or N to the chat? Okay, so that's why, yeah, we're adding a phosphate to our molecule, but this is not a transferase. This is an oxidoreductase. The other kind of clue for that is, well, we've got our electron carriers, NAD and NADH. And then that name, dehydrogenase. A dehydrogenase is a commonly seen in a oxidoreductase. Okay, so what we're going to do, though, is we're going to add this phosphate onto glyceraldehyde 3-phosphate. We make this doubly phosphorylated molecule right here, 1,3-bisphosphoglycolate, and then phosphoglycerate kinase comes in. And what it does is, it's going to take the phosphate from this molecule where I've got my star, and it's going to add it on to ADP to make ATP. So let me ask you this. Give me a 7 in the chat if you think that this is an oxidoreductase, and give me a Z in the chat if you think that this is a transferase, this phosphoglycerate kinase. Z for transferase, a 7 in the chat for an oxidoreductase. Absolutely. It's a transferase because we are transferring this phosphate, the one that is in this red phosphate that I've put a circle and now a square around, transferring it onto ADP to make ATP. So another way to think about that is whenever you see kinase, I want you to think, aha, transferase. Give me a Y or an N if that sounds good. Okay. Now, just to kind of rewind for a second, how many of this molecule, this glyceraldehyde 3-phosphate, how many of that molecule are in the cell at this point in time from our original glucose? How many have come from that original glucose? And I'll give you some options. Is it 0, 1, or 2? Or we'll go up to 3. Sure. My question was, how many of these molecules are we working with dating back to whenever we started with our 6 carbons from glucose? Ella, spot on. We have 2 of these. Could everyone give me a 9 in the chat if that makes sense? And where that number is coming from. Okay. Now, since there's 2 of this glyceraldehyde 3-phosphate, how many NADH have we made? 2. Then how many ADP did we need? 2. And now, last but not least, how many ATP have we made? Do we have now? Or yeah, how many have we made? Perfect. We've got 2 ATP. So if you think about this in terms of investing, we invested 2 ATP. How many ATP have we returned so far? It's okay. We've returned 2. So if you think about it in terms of like debt and payback, we're even. Give me a minus sign if that makes sense. Okay. Okay, great. So at this point in time, after phosphoglycerate kinase, we're even. We invested 2 ATP in the first half of glycolysis, and now we've gotten 2 ATP back. Okay, I got a private message question. For step 5, there is a GAP and DHAP. DHAP goes through a reaction to get GAP, and nothing happens to the original GAP to get 2 GAP right. Totally. Then that was qualified with, sorry, I hope that makes sense. That makes perfect sense. Yes, I'm happy that you asked that because I'm guessing that you're not the only person who had that question. If we go back to, I like this shot right here. So here what we did was we took our fructose 1,6-bisphosphate and we split it into glyceraldehyde 3-phosphate and GAP. or sorry, glyceraldehyde 3-phosphate and DHAP. This one right here, nothing has to happen to it. That's what we needed. And so this one just sort of, it's like these three carbons, they just sort of chill out and they're like, okay, what we need now is to use triose phosphate isomerase to make another GAP. And once we now have two GAPs, then we can proceed. So you could kind of look at this and say, well, these three carbons right here, they never touch the enzyme triose phosphate isomerase because they don't need to be isomerized. I hope that makes sense for everyone. I hope that clears some things up. So where we are right now, to bring it back to this slide right here, is we just completed the step that gave us our two molecules of 3-phosphoglycerate. So we just got to this point. So we have collected our 2 ATP. We've also produced 2 NADH. So we're making progress on this. So now what we need to do is look at this. We've got a mutase, and then we have an enolase. Those are our two enzymes: phosphoglycerate mutase, enolase, and then we're all the way home with pyruvate kinase. So this is where, okay, so we just got to this point where we are now even. We are good to go. Phosphoglycerate kinase made 2 ATP molecules. We've got these 2 molecules of 3-phosphoglycerate. So now we can proceed. 3-phosphoglycerate, how would you classify this enzyme? And I'm going to go atom by atom. We've got a carbonyl carbon with an oxygen. Done, done, same. Negative oxygen, done. Hydrogen, hydrogen. Carbon 2, OH, I'll come back to that. Hydrogen, hydrogen. Carbon 3, hydrogen. Then we got a phosphate. Oh, and then we got carbon 3, hydrogen. Oh, there's my OH group. Check, check. And then here's my phosphate. Check, check. Oh, those look, I think that, how would you, yeah, it's an isomerase. Okay, it's known as a mutase, but that has to do with the mechanism. That has to do with how the different atoms and electrons are moved around. We're not getting into that for this class, but this is an isomerase. So basically what we did was we used an oxidoreductase to make NADH. Makes sense. Then we used the transferase to make ATP. Okay. Then we rearranged the atoms. Then what we did was we've got this 2-phosphoglycerate. Our 2-phosphoglycerate right here is going to be used to then remove an H and an OH to produce water. That gives us this high energy compound known as phosphoenol pyruvate. Phosphoenol pyruvate is basically our second to last step right before what we want, and that is pyruvate. So when we remove water from 2-phosphoglycerate, we make phosphoenol pyruvate. Phosphoenol pyruvate in a singular direction is acted on by the enzyme pyruvate kinase, which is how do you think you want to classify that enzyme? We've got a phosphate here, and then if we look at pyruvate, I don't see a phosphate on pyruvate. Other reactants, I see ADP, and then I see another product of ATP. How do you think you would want to classify pyruvate kinase? Maybe as this phosphate gets transferred to there. So maybe a transferase. Perfect. Okay, now follow-up question. How many of these do we have? How many phosphoenol pyruvates? And just a little bit of a reminder, when we were back here, we knew that we had 2 glyceraldehyde 3-phosphates, which were used to make 2 NADH, and 2 1,3-bisphosphoglycolates, which were used to make 2 ATPs and 2 3-phosphoglycerates. Our two 3-phosphoglycerates were used to make two phosphoglycerates. Two phosphoglycerates were used to make two phosphoenol pyruvates and two H2Os. And from that, our two phosphoenol pyruvates were used to make our two pyruvates. At this step right here, this is where we get our, I'm going to call it profit. Profit. Our ATP profit. Because we paid back quite a ways back. Phosphoglycerate kinase got us even on our ATPs. Pyruvate kinase gives us our ATP profits. Oh. Now, give me a 1 is, oh, this is easy. I got it. A 2 is, I'm going to spend some more time on this. A 3 is, I give up. And you can send it as a private message. Give me a response to that. 1.7. I like it. That's good. That's a good number. I think that this is definitely something you want to spend time with. I think that the more that you break it down, the more consumable it is. Like this is a 72-ounce steak. You're not going to pick it up and just bite right into it. You're going to cut a piece off. Okay, I feel good about this piece. The more that you can simplify the reactions, the better off you're going to be. Now, with that, I want to bring up another point. There are a total of 3 steps that are irreversible. Those 3 steps are hexokinase, phosphofructokinase, pyruvate kinase. All 3 of those steps are transferase reactions. Oh, that's interesting. Yeah. Now, one thing that I would like to add is just because a step is a transferase does not mean that it's necessarily irreversible. So don't get these two things mixed up. Just remember, okay, well, I've got 3 irreversible steps. These first 2 right here are in my investment. This is my last step. So I've got my 2 of my irreversible steps are in my investment phase. One of them is in my return, and one of them is like my most important return. There are several kinases: hexokinase, phosphofructokinase, and pyruvate kinase. 3 of these 4 are irreversible. Phosphoglycerate kinase is a reversible step. Now, this is just, okay, let's take this information and process it a little bit more and maybe categorize it in a slightly different way. What I'd like to also bring up, I've got basically 3 more things that I want to bring up. This enzyme hexokinase, that's our first step in glycolysis. Glucokinase does the same thing. The difference is that glucokinase is uniquely found in the liver. Hexokinase is basically everywhere. So every cell is going to be taking glucose and breaking it down. The liver is a little bit of a different animal, though, because the important thing about the liver is that it stores excess glucose, but it doesn't compete with other tissues. So the liver is such an important regulatory organ in terms of managing your blood glucose level. Of course, it works with the pancreas in order to produce insulin and glucagon, but the liver is unique. Now, that makes glucokinase also unique. It only acts on glucose. It has a very high capacity. It has a Km for glucose of approximately 10 millimolar. Now, the difference between hexokinase and glucokinase, one difference is that, well, hexokinase is an enzyme that can takes on any hexose, any 6-carbon sugar. like glucose, fructose, mannose. Glucokinase says, I only want glucose. Now, hexokinase has a Km of about zero point 0.1 millimolar. Between glucokinase and hexokinase, add an H for hexokinase and a G for glucokinase. Which one of these two sugar or which one of these two enzymes has a greater affinity for glucose? Greater affinity. A greater affinity comes with a lower Km. So hexokinase with a Km of approximately zero point 0.1 millimolar compared to glucokinases, which is 10 millimolar. So basically what that means is that if you gave hexokinase and glucokinase, hey, here's a concentration of, or here's sugar, here's glucose with a concentration of 1 millimolar. Hexokinase is going to be like, wonderful. Glucokinase is going to be like, okay, cool. Listen, I'm going to be most responsive whenever sugar levels are high because that's what I do. That's me. That's glucokinase. I'm a liver enzyme. Now, your blood glucose level is usually around 5 millimolar. When it gets higher than that, then glucokinase is like, whoa, whoa, we got a high blood sugar. We got to do something about this. So, what I want you to know about these two enzymes is specifically their organ distribution. Well, glucokinase is uniquely in the liver, hexokinase is basically everywhere. And you want hexokinase to have that high affinity or that low Km because then when hexokinase is in every different type of tissue, it can grab onto sugar and break it down, grab onto glucose and break it down. Okay, now with all of that said, here's your summary of everything. And this is a big this is a lot to process. But one glucose is going to give you a net of 2 ATP and 2 NADH. That ATP, could anyone tell me in the chat, where does ATP come from? What organ? Or what organelle, I'm sorry. I'll give you a hint, it's the powerhouse of the cell. Mitochondria, powerhouse of the cell, that's right. Um, yeah, we're going to produce ATP, and that's your energy currency, which is great. But this NADH, that's kind of the more important one. I always kind of think about it like ATP is like, for some reason, this has just stuck with me over the years. Um, ATP is like having a lot of money in your wallet or your purse or whatever, wherever you carry your money. If you have a lot of ATP in your wallet, people are going to be like, dang, you're rich. Um, but really, that just means you've got a lot of money in your wallet. NADH, by comparison, is like, oh, I've got a financial portfolio. That, by comparison, I'll say, and you're rich if you've got financial portfolio and lots of cash in your pocket. Um, I'm going to say cash in pocket. This having the ATP, having the cash in your pocket is like, yes, I'm going to Wendy's. What's up? Whereas the financial portfolio wealth is like, I'm going to buy all of the Wendy's. So that's the comparison. But the point is that this NADH gives so much more energy. And the way that you get so much more energy from ATP is with this right here, the electron transport chain, also known as ETC. The point of the electron transport chain is to take this high energy electron carrier, NADH, and move some electrons. And we'll go into this more later, but it's moving electrons from all of these different protein complexes in the process, pumping protons to one side of a membrane outside of the mitochondria. And then we're going to generate a huge amount of ATP. So, with that in mind, like, you don't get most of your ATP from glucose just by doing glycolysis. Instead, you get it in kind of a secondary way by producing NADH and FADH2 and other high energy compounds. Could you give me a NADH in the chat if that makes sense? Okay. Okay. I am going to go ahead and stop the recording. We covered a lot of stuff today. If you got any questions,